Hi there, this is another biochemistry session on polysaccharides and specifically homopolysaccharides. So polysaccharides, they differ in the chain length, the type of the monosaccharide units that they're made up of, the degree of branching that they have, and the type of glycosidic bonds between the monosaccharides. So homopolysaccharides are polysaccharides that are made up of a single type of monosaccharide unit. Heteropolysaccharides are polysaccharides that are made up of two or more different monomer types, and both can be branched or unbranched. So can you come up with two examples of homopolysaccharides that you have learned before? I'm going to reveal the answer. Starch, cellulose, and glycogen are some of the possible answers. They're all made up of only glucose monomers. Functions of homopolysaccharides include storage, starch in plant cells and glycogen in animal cells, and also structural support, cellulose in plant cell walls and chitin in fungal cell walls, as well as animal exoskeletons. Functions of heteropolysaccharides include extracellular support, so that will be peptidoglycan in bacterial cell envelopes, and proteoglycan in animal cell extracellular matrices. Unlike proteins, polysaccharides do not have defined lengths. This is because proteins are synthesized from mRNA templates, which have a fixed length, but polysaccharide synthesis depends on only the enzymes that catalyzes the polymerization. Homopolysaccharides for storage, that would be starch and glycogen. So for starch, we have amylose, which is linear, and it only has alpha 1 to 4 glycosidic bonds. If you don't know what this means, please visit the disaccharide video. For amylopectin, it includes not only the alpha 1 to 4 glycosidic bond, but also the alpha 1 to 6 bonds, which allows for branching points. And thus, amylopectin is branched. For glycogen, it has the same glycosidic bonds as amylopectin, but it is more extensively branched because it has more frequent branching points along its linear chain. So image A shows the a linear strand of amylose. Image B shows the branching points in amylopectin or glycogen. They're the same. Image C shows what starch looks like in starch granules. As you can see here, the amylopectin forms a lot of double helices with itself, or it can form double helices with amylose. Sorry, I was meant to say that amylopectin forms a lot of double helices with itself and also with amylose. And as you can see here, amylopectin has a lot of branches and thus a lot of non-reducing ends. The same case goes for glycogen. And because enzymes remove glucose from the non-reducing ends, when energy is needed, the multiple branching points lead to more non-reducing end and thus allows enzymes to break glucose from these multiple non-reducing ends simultaneously, allowing for a faster mobilization of glucose residues. So structure fits function. Why don't cells just store glucose monomers instead of glucose polymers? This is because the concentration of glycogen molecules is lower than the concentration of equivalent glucose molecules, right? Because 1 mole of glycogen molecules will be like, 1,000 moles, for example, of glucose monomers, right? If you break all of its monomers apart. So because it has a lower concentration, it decreases the osmolarity pressure and thus prevents the cell from lysing due to hypotonicity. So take a moment to pause and digest the information. All right, we'll move on. So now we're going to talk about homopolysaccharides that serve structural roles. That will be chitin and cellulose. So for cellulose, it has beta 1 to 4 glycosidic bonds because cellulose are made up of beta glucose monomers, which is different from amylose, which is made up of alpha glucose monomers. And thus, this glycosidic linkage causes individual molecules of cellulose to fold differently from amylose in space, and thus leading to different physical properties. And chitin also has this beta 1 to 4 glycosidic linkage, and it also has a linear form, though its monomers are made up of N-acetylglucosamine, and that so chitin is basically like very similar to cellulose. The only difference is that the alcohol group on the number 2 carbon in cellulose is replaced with acetylated amino groups. And this replacement makes it more hydrophobic and water resistant than cellulose. And thus, chitin exists in the exoskeleton of many animals. Steric factors and hydrogen bonding influence the homopolysaccharide folding. So let's first look at a disaccharide, right? In this glycosidic linkage, you have this phi torsal angle. So that is between the anomeric carbon and the oxygen. And then you also have the, is it psi torsal angle between oxygen and the number 4th carbon. And in principle, because they are single bonds, they can rotate freely, right? The range for each bond will be between 0 degrees and 360 degrees. But due to steric hindrance, because at certain conformations, the two hexagons are going to clash with each other, as well as hydrogen bond stabilization, meaning that at certain conformations, the hydrogen bonds will be able to stabilize and cause like intramolecular interactions. Due to these two properties, there is a set conformation for each polysaccharide, and thus leading to different macrostructures. So if we look at a disaccharide here, this low energy conformation is extended to minimize steric hindrance from the two hexagons, or I shall say glucose monomers. Sorry, actually in this photo it shows galactose monomers. But anyways, it doesn't really matter in this case. And it also maximizes hydrogen bonding so that it stabilizes the structure. But at high energy conformation, these two monomers are clashing with each other and thus is sterically hindered. So there is only, some ways of rotation that is possible at these two sites. So it turns out that for amylose, this rotation will lead to a helix. So the angle between the planes of each monomer is 60 degrees in amylose, and thus there's 60 monomers per turn of the helix. And for amylopectin, it's basically the same. The only differing part is that it has this alpha 1 to 6 glycosidic linkage. For cellulose, each monomer is turned 180 degrees relative to its neighbors. So you'll see that, the monomers are sort of upside down to each other. They're flipped per unit. And this causes the interchain as well as intra-chain hydrogen bonds, which produces a very stabilized structure, and that's why it provides support for plant cell walls.